Hello. Uh, this video presentation has been designed to supplement the PowerPoint presentation that is posted in Canvas. You'll notice that the PowerPoint presentation contains a bunch of facts that I'd ask you to read over and review. And I'm really going to focus this presentation on some of the more complicated physiology and and chemical and biological principles that are really important in our cells. So this a lot of this lecture, at least the beginning of it, focuses on how molecules move across the membrane to get inside or outside of our cells. So sometimes molecules do not need carriers, and this would include, for example, the simple diffusion of lipid-soluble molecules. It also can include the simple diffusion of ions through nonspecific ion channels that are open and existing in the membranes. Uh, Also, the simple diffusion of water, which we call osmosis, and this occurs through movement of water through aquaporin channels. And we'll also look at some examples of carrier-mediated transport. This includes facilitated diffusion and active transport. So the transport can be passive or active. So passive transport involves molecules moving from a higher concentration to a lower concentration. So we call a concentration gradient the difference between the concentration of the molecule on each side of the membrane, right? So we know if a molecule is more concentrated on one side of a membrane than the other, it's going to want to move to the less concentrated side to be in equilibrium to have the same concentration on both sides of the membrane. Active transport occurs when molecules are being moved in a manner opposite that of the concentration gradient. So energy is needed to move a molecule from a lower concentration area to a higher concentration area, and this is going to require ATP and very specific carrier pumps. So let's take a closer look at some of the mechanisms of passive transport. So in A, over here, we have some nonpolar molecules, and these nonpolar molecules can freely diffuse across the membrane following simple diffusion. Likewise, if we have channel proteins that allow, for example, ions to be crossing the membrane, these ions will be moving across the membrane via simple diffusion from a higher concentration to a lower concentration in this mechanism to obtain equilibrium in the concentration of this molecule in both sides of the membrane. And then in the bottom, we see carrier proteins, which are bigger, and they really need a protein to facilitate its entry, where they can enter the carrier protein, and then the carrier protein actually needs to change its conformation so the molecule can get across the plasma membrane. And again, this is passive, and really simple. The diffusion kinetics of moving from a higher concentration to a lower concentration are catalyzing this process. So what I'm showing you now is just this principle of diffusion. And here we have a molecule in high concentration on the left and in low concentration on the right. And we see with the little black dots indicating the concentration of the molecule and the number of particles. So diffusion is going to want to make there'll be equal concentration on each side of this barrier, right? Shown in the middle with that dotted line, right? So if you have a high concentration over here or low concentration there, diffusion is going to make it be equal on both sides of the barrier, the permeable barrier, right? So when we say net diffusion is occurring, that means here we're going to have a net movement of more molecules moving from the left compartment to the right compartment. Once there is an equilibrium established, yes, there'll be some movement of molecules from the right compartment to the left compartment and some from the left compartment to the right compartment, but there'll be no net movement because overall the numbers are going to stay similar between the two compartments. So I just want to be clear that even when there's equilibrium, there is movement. However, there's no net movement, meaning that things are going to be staying relatively constant between the two compartments. Again, the critical thing is that in these compartments, that this barrier is allowing for the movement of these particles between the two compartments. One example in our cells where we have complete movement of a molecule across our plasma membranes is with gases. So we see this with, for example, oxygen and carbon dioxide. So here we see, for example, this could be in the lung, where we have a high level of oxygen in the air we're breathing in. And basically, the oxygen will go freely pass into cells, for example, red blood cells, and they can then pick up the oxygen molecule as they're passing through the capillaries in the lungs. But then imagine, let's look at some red blood cells that just traveled through an exercising muscle. In that case, they may have given up their oxygen to give it to the muscle cells, and the muscle cells are releasing carbon dioxide from their active metabolism, and then the red blood cells could pick up carbon dioxide, and then when the red blood cell gets to the lungs, then it can then dump out the carbon dioxide, and it'll freely diffuse out because of the concentration gradient. And then, of course, that allows us to exhale the carbon dioxide, and we'll be learning more about this later in the semester in greater detail. So I just want to show you some ion channels, a little bit with a closer look. Here's the cytoplasm on the left, extracellular fluid on the right. The ions are shown with the green balls. And here is a closed channel. So when the channel is closed, the ion cannot cross the plasma membrane. However, when the ion channels are open, simple diffusion will allow ions to be traveling across the plasma membrane. So osmosis is the movement of water across a semi-permeable membrane. And in this situation, what I'm going to show up here, here's a container with a semi-permeable membrane. Here we have a solute depicted with purple balls. So you probably notice that there's more solute in the right compartment than the left compartment. And the semi-permeable membrane will not allow these solute balls to be passing through the two compartments. But what can pass through is water, because the holes are small enough for water, but not small enough for these bigger molecules to pass through. So with diffusion, we know that nature wants to have everything in equilibrium. So we want to have equal concentrations of a solute or a molecule on both sides of a semi-permeable membrane. So in this case, because the solute cannot move, what can move is the water. So what is going to happen is the water is going to end up moving from this left compartment to the right compartment. It's going to do it so that basically the concentration of particles in the left compartment will be equivalent to the concentration of particles in the right compartment. The right compartment will just have a bigger volume, and the left compartment, after osmosis, is going to have a smaller volume. But what will happen after the osmotic process is that we'll have a similar concentration of solutes, for example, like 10 per mil in the left compartment and 10 per mil in the right compartment after osmosis has occurred. And we're really going to be seeing the net movement of the water, again, from the left compartment to the right compartment to make things, to make the concentration of solute per volume of water equal. So let's take a look at a osmosis experiment in a beaker in the lab. So what we're seeing here is that there's a balloon that has the semi-permeable membrane allowing water to pass through the membrane but not sucrose. And this ball, or balloon ball, has 360 grams per liter of sucrose in it. Then in the beaker, we notice that there's 180 grams of sucrose per liter of sucrose in the water in the beaker. So there are more solutes, which is sucrose, inside the balloon than outside of the balloon. So because we know that what nature would like to do would be to basically have there be equal amounts of sucrose both inside the ball and outside the ball. But since the semi-permeable membrane doesn't allow the movement of sucrose, what is going to happen is the water is going to move to the inside of the balloon. The balloon is going to grow in size, as depicted by those little halos around the balloon. The balloon will be growing in size, having more water inside of it. And then there's going to be less water inside of the beaker. So if you add these two numbers together, 360 plus 180, you get 540 grams per liter. And if you divide it by 2, you get 270 grams per liter of sucrose. So osmosis will stop when the concentration of sucrose inside the balloon is 270 grams per liter sucrose and 270 grams per liter sucrose in the beaker, right? And in order for that to happen, the sucrose in the balloon needs to be diluted to have it be going from 360 to 270 grams per liter. And then we have to lose water in the beaker so that the concentration of sucrose will be increased to 270 grams per liter. So when I look at this picture, it reminds me of some phrases that we've all heard of in other bio classes. And the terms I'm thinking of are hypertonic, hypotonic, and isotonic. So when you look at this picture, as shown now, you see that the sucrose balloon is hypertonic compared to the water in the beaker, right? Because there's more solute inside the balloon than inside the beaker. The sucrose solution in the beaker is hypotonic compared to the balloon, meaning there's less solute concentration inside the beaker sucrose solution than inside the balloon sucrose solution. And after osmosis occurs, where the balloon is going to grow in size by bringing water in, and the water volume inside the beaker is going to reduce, right, as the water has moved onto the balloon. After osmosis has completed, there's going to be an equilibrium where there's going to be 270 grams per liter sucrose inside the balloon and 270 grams per liter sucrose in the beaker. That will be two isotonic solutions, because the concentration of solute is going to be the same between the two solutions, the solution inside the balloon and the solution inside the beaker.